All right, welcome back. This is the last lecture on Chapter 2 on the chemistry of life or body chemistry. Last time we were working our way through molecules, both inorganic and organic. We had started off talking about carbohydrates, then we talked about lipids, and now we're going to finish off with the last two categories, proteins and nucleic acids. Proteins and nucleic acids. All right, so proteins, okay? Proteins is the third category that we're talking about. It is organic because it contains hydrocarbons. And proteins have a very unique and important place within life because I like to call them the Swiss Army knife of biomolecules. That is, if you're familiar with a Swiss Army knife, it has lots of little doodads that can bend out and have different functions, like a corkscrew or a saw or a knife or a scissors or a toothpick. Okay? The amino acids, which compose proteins, are like that. They have different functional groups that have different roles. Okay? So proteins are very diverse in their function. Okay? Proteins are the most abundant and important organic molecules in the body. They contain basic elements that we've already talked about, C, H, O, and N, and they are composed of these 20 amino acids. Okay, there's 20 amino acids that form proteins. So there are at least 7 roles or functions for proteins in the body. Okay, this is just in the human body or in animals in general. Support, movement, transport, buffering, metabolic regulation, coordination, and signaling, and defense. Okay? In the next 7 slides, I'm going to go through a couple examples that I want you to know. Okay? Proteins are so important. When someone would ask you the question, what is the most important protein in the body? Okay? And you could come up with a couple different answers. As long as you give a good rationale, you could have a pretty good answer for any of them because they're all mission critical. Like if you missed any of them, you would die. You would die for different reasons, but you would still die. All right, so let's visit a couple of these examples of important proteins in the human body that have very different roles. Okay? They're not all just enzymes. They're not all structural. They're not all antibodies or used in defense. They have different roles. Okay, so collagen. Okay, collagen is the most abundant protein in the human body. Okay, by far. Collagen, type 1 collagen. There are actually up to like 20 different family members of collagen. But the type 1 collagen is the most abundant. It's found in our skin, in the epidermis, in our hair, in our cartilages, okay, the joints of our bones, our derma, the connective tissue that holds us together, is just chock full of collagen. Okay, it is a tripeptide and is a very important fibrous protein. Okay, it holds our body together and gives us durability. Okay, next major role for proteins in the body is movement. Okay, this is a portion from the muscle chapter, muscle biology. You can see the bicep here. If you look closer and closer and closer, eventually we get on the inside of a single muscle cell and we see our good friend actin and myosin. Some of you may have heard of those proteins before. Actin and myosin lie next to each other, and myosin reaches over and grabs actin and pulls on it, and that is the basis for muscle contraction. Okay? So producing movement, these two proteins interacting is the basis of body movement. Okay, so proteins cause movement. Okay, next category here. Okay, this is actually a molecule of albumin. Okay, it's also shown here in green, but albumin is a secreted protein in our blood, and it circulates in our blood, and it's a carrier protein. It's like a taxi cab. Okay, it floats around in our blood, and it carries molecules around. Molecules will hop on, ride for a bit, and then hop off. Very similar to another protein that you're probably more familiar with, hemoglobin. Hemoglobin is a globular protein. It is found in our red blood cells. It is also like a taxi cab. Okay? Oxygen hops on in the lungs and then travels around the body, let's say to the muscle, and then hops off. Okay? The taxi cab never leaves the blood, okay, the hemoglobin. So it's a carrier molecule. It carries things around. So that's another key role for proteins in the body. Okay. Another one here is a buffering system. There's all kinds of proteins and peptides that help to buffer our blood. Okay, and so those are important for as chemical buffers. Proteins serve that role. Okay, metabolic control. Okay, so the enzymes that control the different transformations between different substrates and products, okay, are controlled by enzymes. And so metabolic control using enzymes is very important, and it's one way that proteins play an important role in our bodies. So all of our cells have lots of enzymes that can move a chemical through a series of reactions to change from substrate into product. Okay? You can think about glucose moving through pyruvate and finally forming some, at the end of that you would form ATP and you would form carbon dioxide as a waste product. Okay? But the important thing to realize here is that these purple ovals here are individual enzymes that are acting kind of like an assembly line. Okay, in an assembly line, you're in a factory and the product is moving in front of you, and each member of the assembly line is doing one special addition or action to that product, and then it moves down, and then the next worker does something else. That's kind of like how enzymes work within our cells. Okay? Enzymes are proteins. Okay? Some of the most important enzymatic reactions are involved in redox reactions for converting glucose and oxygen into CO2, water, and waste. Okay? This is glycolysis, Krebs cycle, and then oxidative phosphorylation in the electron transport chain. Okay? So lots and lots of reactions here that you will learn if you take biochemistry. Another role for proteins in the body is signaling. Okay? So communication and signaling, this is done by proteins called hormones. Okay? There are many of our hormones that are proteins. Okay? Some hormones are lipids, so that's another class of hormones. But this is oxytocin, here's growth hormone, and here's FSH, or follicle stimulating hormone. It's very important for reproduction, okay? Reproductive progression. And all of them are traveling around in the blood, and they signal from one part of the body to some downstream target. Okay? So that's a signaling function. These proteins act as a message that is sent and received. Okay? Another role for proteins. Okay? Here's another one. This is an antibody. Okay? We typically we see antibodies illustrated as kind of a Y shape. Okay? Here's that characteristic Y shape. But this is in fact what it looks like in a space-filling model. Okay? Here's the bottom of it, and there's the tips. Okay? There's an antigen binding site here and here. The role of this protein, again, is to float around in the blood or the lymph or the tissue and identify and strongly bind to bad guys, okay, as I like to call them. Okay? Foreign proteins or bacteria or viruses, antibodies can recognize and bind to them with high affinity and tag them for destruction. Okay? So it's a defense mechanism. Proteins are used for defense in our immune system. All right, so those are 7 different roles for proteins. Okay? A very important class of molecules are proteins. So what are proteins? What are they made of? Okay? We have to talk about amino acids. Okay? Proteins are composed of amino acids. Think about them, like I said before, as a pearl necklace. Okay? A pearl necklace has a pearl, and then a pearl, and then a pearl, and they're linked together with a thread or a chain. Okay? And so those pearls are the amino acids, and you string them together in a long chain, and then that pearl necklace actually folds on itself and it forms a globule that has important characteristics. Okay? So here's some of those characteristics. Some of the features of amino acids, but again, a figure is always easier to describe than just words on a page. So this is represents a single amino acid. Okay? There are 3 main chain molecules: a nitrogen, a central carbon, and a carboxylic acid. Okay? And then attached to the central carbon, there's this R group. Okay? And this R group is just, the R represents some variable group. And these can vary. There's actually 20 different types of R groups. Okay? So you could have a long chain, you could have a short, just a carbon or just a hydrogen. You could have a cyclic form here. There's lots of variety in the different amino acids. But every amino acid has an amine group and it has a carboxylic acid group. And thus the name amino acid. Amino acid because it has an amine and a carboxylic acid. Okay? And so these are the groups that serve as the linkage points to the next door neighbor amino acid. And this R group is kind of like a charm bracelet. I don't know if those are even popular at all, but if you don't know what a charm bracelet is, you have little links of chain that wrap around the wrist, and then sticking off at different angles are these little charms or little trinkets or shapes of a teddy bear or the world or your boo or whatever. I don't know. Okay, that's what the R groups are. So we hook together those amino acids using what's called a peptide bond. Okay, again, let's look at an image. Okay, here's amino acid number 1. In this case, it's glycine because it's a single H. And here's alanine because the R group is a carbon. Okay, I'm not going to ask you to memorize them. You have to memorize them for biochemistry, but not for my class. Okay, we have 2 neighboring ones, and this OH group of the carboxylic acid and the H here of the amine. If we have performed dehydration synthesis, now we've formed a peptide bond. Okay? It's a covalent bond that links the 2 amino acids. A very, very strong bond that only can be broken through a hydrolysis reaction. But under normal circumstances, this is a very strong, it's a covalent bond. Okay? Now, linking these amino acids into a long chain is what we call primary structure. Okay? Primary structure. That's this first level of understanding how proteins are assembled and how they achieve their ultimate function. Okay? You have potentially four levels of structure. Okay? Primary, secondary, tertiary, and quaternary. And what I just talked about was the primary. That is the sequential linking of those amino acids in a linear chain. Okay? We've not yet folded, we've only connected them in a chain. Okay, so here's the diagram that gets at all four of these types of protein structure. So here illustrated is the primary structure. And in this secondary structure, we begin to fold or form these alpha helices or beta sheets. Whether we form alpha helices or beta sheets is completely dependent on the nature of these R groups. But they begin to take not just a two-dimensional linear structure, but actually a three-dimensional structure. Okay? Alpha helices and beta sheets are examples of secondary structure. Once we've formed alpha helices and beta sheets, well then this alpha helix and this alpha helix will begin to associate and form some interhelical interaction. And then together that forms what we call the tertiary structure. Okay? One alpha helix or one beta sheet is an example of secondary structure. But if you take a combination of a couple alpha helices and a couple beta sheets and kind of mush them together, now you've achieved tertiary structure, okay, third order. Some proteins only achieve tertiary structure. That is because they only consist of a single peptide chain. However, some proteins, mature functioning proteins, are more than just a single chain. For example, hemoglobin. Hemoglobin is the carrier protein that we just talked about. It exists in the red blood cells. It carries around oxygen. Hemoglobin actually consists of four distinct peptide chains. That is, there are four, let's say, pearls on a string, pearl necklaces, that the four of them kind of come together, and purple and purple and blue, there's four of them. That is now quaternary structure. Okay? So to achieve quaternary structure, you need separate individual peptide chains to then associate with each other in a stable form. Okay? Primary, secondary, tertiary, quaternary. If a protein does not achieve its correct shape, it will not possess function. Okay? So protein folding is very important. And all the chemical principal properties that we talked about with hydrophilic and hydrophobic are the determining factors for achieving the correct folding. Okay? That's, again, a lot of biochemistry underlies that. There are two structural classes of proteins. You can have fibrous proteins and globular proteins. Again, let's take a look. Okay? Here's, again, hemoglobin. It's a globular protein. That is, it resembles kind of like a ball of string. Okay? The ball of string is kind of jumbled up, and the folding of the helices and the beta sheets makes that globular structure active. Okay? And the precise binding location of the heme unit here with the iron that is coordinated there allows for oxygen binding. And there's all kinds of important molecular dynamics that govern the affinity of oxygen for that heme group. But that's a globular protein, okay, like an enzyme. Here's a fibrous protein, like collagen. Okay? Collagen is a triple helix, and it's like a braided rope, okay? Because its function is more structural. It provides resistance to tension. So keratin in your skin and collagen and elastin, those are fibrous proteins. They look kind of like a braided rope because they're more structural rather than kind of like enzymatic or receptors or hormones. All right. So enzymes, okay? Enzymes are one of the 7 major functions that I described as far as roles of proteins in the body. But enzymes are massively important. If enzymes don't catalyze chemical reactions, we would die. We talked about carbonic anhydrase catalyzing the transformation of Ch.2 into bicarbonate in the blood. Okay, that's the role of enzymes. Okay. Enzymes have high specificity, they have saturation limits, and they are highly regulated. That is, we can turn them off or on depending on binding and other regulatory elements in the enzyme. Okay. But an enzyme catalyzes or speeds up some reaction. Okay? What is a reaction? It's bond making or bond breaking. All reactions are some type of making a bond or breaking a bond, usually a covalent bond. Enzymes speed up that bond making or breaking. Okay, so here's an example. Substrate 1 and substrate 2. This is going to be what type of reaction? Okay, it's going to be a synthesis reaction. The enzyme has some specially shaped parts of the enzyme that will recognize substrate 2 and will recognize and have a high affinity with substrate 1. And because they're both attracted there, oh, what do you know? They're going to join, and actually S1 is going to be joined to S2. If you think in this way a little bit as like the Baptist church matchmaker, I don't know if the example is going to help you, but there's always that lady that sits in the back of the church and kind of looks at all of the young people that are in church like, oh, little Susie would make a good match for little Bobby, okay, or whatever. She's matchmaking. She knows them both. She's good friends with them both. Say, hey, Bobby, do you know my friend Susie? I'm going to introduce you to each other. I know Susie and I know Bobby, but you guys might not know each other, okay? And so the matchmaker attracts and then joins, and then they go off and they get married, whatever, okay? But enzymes are kind of like a matchmaker. They have relationships with individuals, and then through that mutual interaction, some other chemistry can happen. You get my drift. Okay, there we go. Now, once they're both bound to the enzyme in the proper orientation, they can interact and form a covalent bond, okay, if it were. There's a product, okay, and the product can float away, and little Miss Matchmaker can go on and do it again with another set of substrates, okay? So an important concept for enzymes is they don't get consumed in the process. They get reset and they're ready to go for another round of catalysis. Proteins also can function as cofactors and coenzymes, and there's isozymes. Those are concepts that we're not going to get into, but you should, if you come across these terms later on in the course, you'll know that they're proteins that play a role in catalysis. They're helpers, you could say. Important concept. We talked about how proteins can fold and achieve their mature and proper functioning. And there are many factors that influence protein folding. Some of them are temperature and pH. Those are really the big ones. Okay? There are other chemical things that can cause denaturation, but temperature and pH changes are really the ones that I want you to think about. Okay? If we have high changes in temperature or we have high fluctuations in pH, proteins don't like that. Okay? Proteins will fall apart and they'll denature. Okay? If you think about an egg, okay, if you fry an egg, you're denaturing the albumin. Okay? If you crack the egg into the frying pan, that high heat denatures the protein and it gets fried. There's no way to unfry an egg. Okay? It's totally toast, like it's gone. It cannot be returned back to its original state. Okay? So protein denaturation destroys, in most cases, it will destroy the function. There are some proteins that exist in our bodies that are tremendously robust. You can denature them and then put them back into a cell or into the body, and they'll reform just as they were before. Okay, but that's rare. Okay, glycoproteins and proteoglycans. These are proteins and sugars that kind of have that are bonded to each other and have unique roles. We will talk about these when we talk about joints and synovial fluid and other components. Okay, so if you look at this word here, glyco, like glycolysis, means glucose. Okay, so sugar. Protein, so sugar protein. Okay, it's a sugar modified protein. And here's just the reverse. Proteo, that's like protein. Glycan. Okay, so it's a protein modified sugar. It's mostly sugar with a little bit of protein. This is mostly protein with a little bit of sugar. So the back of the word is the major part of it, and this is really describing what kind of protein it is. All right, last category, okay, or organic molecule. We've gone through carbohydrates, we've gone through lipids, we've gone through proteins. Okay, and proteins we spent a lot of time because of all the diverse roles that proteins play in the body. Okay, last one, nucleic acids. Okay, the fourth of the four categories of organic molecules. Okay, and you'll probably recognize this as important for our genome, okay, our DNA. Deoxyribonucleic acid, okay, is DNA. Okay? The deoxy part is the important. We'll look at the structure in just a little bit. That's really the difference between DNA and RNA. There's a couple differences, but deoxy is the distinguisher here for DNA versus RNA. So nucleic acids have this general architecture, okay? There's a ribose sugar, there's a phosphate, and then there's a nitrogenous base, okay? Nucleic acids have these three components, okay? One nucleic acid. A sugar, it's usually a ribose sugar, that is a 5-membered ring. There's a phosphate group, that's a phosphorus with 4 oxygens attached to it. And then there's a nitrogenous base, okay? That's hanging out here. It's usually a ring structure, sometimes a double ring, with other molecules, functional groups that can interact in other ways. Okay? But you need the 3 parts. Okay? And depending on this nitrogenous base, I say nitrogenous base because there's usually groups here that can be dissociated as acids and bases. And there's usually nitrogens that are punctuating this ring structure. So we call it a nitrogenous base because the nitrogen is present. Depending on what that looks like, it might not look exactly like this one, and it might look like this or this. Okay? These are adenine and guanine. These are purines. Okay? Purines always have a double ring structure, okay? It's kind of the weird way that I remembered. Purine, AG is for awesome God. Awesome God is pure, like holy. I don't know if that's going to make sense to you. AG, awesome God, pure, purine. And then we have pyrimidines, okay? Pyrimidines are the single ring structures, okay? And you have cytosine and thymine, and I just remember they're the other ones, okay? AG and then CT. Now, uracil is unique because it is only present in RNA, okay? T is only present in DNA. So when we're making DNA into RNA, we actually swap out the T and we replace it with the U. So these are interchangeable. It just depends whether you're in the DNA form or the RNA form. But they are completely synonymous as far as the coding. Okay. So DNA and RNA, very, very important for, like I said, encoding our genetic material. There's a polymer, there's a string of nucleic acids that are linked by a covalent bond. And the DNA has a double helix. The RNA has a single chain. Okay. The base pairing rule here that you will learn is that A always bonds with T in the double helix, and C always bonds or base pairs with G in the double helix. And the easy way to remember that is straight goes with straight and curvy goes with curvy. Okay? When I mean straight here, like the straight lines of the A has like straight lines when you're making a capital A, and the straight line of making a capital T. Straight goes with straight, curvy goes with curvy. You make the C, you make the G, those are curvy shaped letters. That's an easy way of memorizing the base pairing. Okay? Uracil, U and T replace each other. Okay, so here's our double helix, double deoxyribonucleic acid. Okay, here's our AT and CG. Okay, they're always, the base pairing there is always C to G, G to C, or A to T, T to A. Okay, and the bonding between these bases is hydrogen bonds. Okay, I mentioned we were talking about how hydrogen bonding is a weak intermolecular force, but because there's many bonds participating in this rung structure, imagine this is a ladder, and the connections in between are the rungs that you would climb up the ladder. It's just been twisted, right? These rungs are stabilized by hydrogen bonds, okay? And if we give it enough heat, an increase in temperature, we can actually melt those bonds, okay? We can actually denature the DNA and cause that ladder to separate. And that's actually what happens during PCR, okay? Polymerase chain reaction. Again, you don't have to know that. I'm just kind of sharing some kind of research stuff. Okay, so ribonucleic acids, deoxyribonucleic acids in this case, okay? It forms this double helix structure, and the code here, the sequence of letters, encodes genes, okay, which will make proteins or make microRNAs or make other downstream products that are important for doing everything that cells and organisms do. It's all encoded in the sequence of the nucleotides. Okay? It's literally a language. It's an instruction book for making humans, okay? Making cells, making proteins. Okay? The backbone here, okay, shown here in this tan color, is the phosphate ribose, phosphate ribose, phosphate ribose backbone. Okay? It's a phosphate backbone, and it's always phosphate ribose, phosphate ribose. One side is the 5' and one side is the 3'. I'm not going to ask you to memorize that, but that's how you determine which end of the backbone that you're dealing with. Because polymerization only happens on one of those, the 3' end. Okay? And then these bases are what interact in the middle of the double helix. Okay? All right, so nucleic acids, very important for encoding genetic information. DNA is the long-term storage molecule. It sticks with us forever. Okay, stays in our nucleus. The RNA, which is transcribed from DNA, is a short-term messenger, okay, message copy of the genetic material. Okay. So these are the types of RNA. That's that short-term message. Okay. You have messenger RNA, mRNA. You have transfer RNA, which is tRNA, and you have ribosomal RNA, which is rRNA. Okay. All of these are involved in what's called translation. That is, there's a paradigm in biology that you really need to know. I'm going to put it up here in this corner. Okay. This is the central dogma. Okay. DNA encodes RNA, and RNA encodes protein. Okay. This step here, okay, of DNA making RNA is called transcription. And this process of going from RNA to proteins is called translation. And the analogy that is useful to use here is because these DNA and RNA are both nucleic acids. Okay? Proteins are proteins. So if this is one language, nucleic acids, and this is the second language, proteins, well, if you're just copying in the same language, we call that transcription. We can transcribe a message because it's using the same language. If we convert from one language to another language, talk about translation. If you're converting English to German or English to Spanish, okay? That's going from RNA, nucleic acid, to proteins. So hopefully that helps you understand. All right, these are the main parts that we just talked about, the components of nucleic acids. Okay, the last thing that we need to mention with regard to nucleic acids is not all nucleic acids are used for information storage in the genome. Some nucleic acids are used for energy. In fact, the chief example, ATP. Okay? Adenosine triphosphate happens to be one of the nucleic acids we use for DNA. Okay? But it also happens to serve another role, which is our energy currency. Okay? The third phosphate bond there contains a lot of energy that we can use for doing work. Okay? So if we're breaking or making that bond, we convert ATP triphosphate to adenosine diphosphate. Okay? And this is the low energy form. This is the high energy form. Okay? So typically when we're making ATP, all we're doing is converting ADP into ATP. Okay? And we're using the energy of reduction and oxidation to move the electrons such that we can phosphorylate ADP. So phosphorylation just means adding a phosphate. Okay? Adding a phosphate is phosphorylation. Dephosphorylating is removing a phosphate. Okay? That's how we release the energy of ATP. So we have an ATPase, that's just an enzyme that can break that bond and then utilize that energy in some reaction. Okay? Because we know that many reactions around the body require energy input to make this reaction favorable. Okay? Hydrolyzing this, this phosphate bond off of ATP releases a buttload of energy. Okay? Pardon my French. Okay? It releases a lot of energy. And we can utilize or harness that energy to an undesirable or unfavorable reaction. Okay? If the enzymes are built right, it does that quite well. So here's our adenosine triphosphate. This is the energy bond here. And we liberate or we hydrolyze that bond, we release a lot of energy. And in the enzyme, there's going to be an ATPase, that is a component of the enzyme that can break that bond, use the energy, and combine it with something else and do some work. Okay. So that is, so you have AMP, monophosphate, diphosphate, and triphosphate. So if you see these, that nomenclature there, AMP, ADP, and ATP. Okay, mono, di, tri. But ATP is the most common energy molecule in all of biology. There's a couple weirdo bacteria or archaea that use a different form, like GDP, I think. But all biology, bacteria, animals, plants, okay, are using ATP. Okay, it's a universally recognized and utilized molecule. So chemicals in cells, we've just done a big overview of chemistry. Okay, we've gone from the fundamentals of matter, energy, atoms, and bond formation down into biochemistry. That is, how do chemical reactions within living things like cells take place? Okay, we need fuels, we need transporters, we need communicators, okay, we need movement. All these biomolecules here, those organic molecules, are so critical for living cells. Okay? And so metabolic turnover, again, we're getting to this concept of the biotransformation of matter and energy. Okay? We can use glucose and oxygen and convert that into ATP. Or we can use some ATP and build a peptide bond or some other matter. Okay, so we can always exchange back and forth depending on what the body needs. Okay, so as we grow, there's always exchanges going on all the time. All right, so here's just a couple tables that go through a review of all of these organic molecules, and up at the top, the review of the inorganic molecules and some things about sources, building blocks, and functions I want to review. All right. This table refers to turnover times. That is, how fast do these molecules turn over in the body? I'm not going to ask you to memorize these right now. We'll talk about them in A&P2 largely. All right. That's about 36 minutes. That concludes our Chapter 2. Okay, so this is a shorter lecture, and so there's going to be some extra time there, but we finished the chapter. The next chapter will be Chapter 3 on cell biology and a review of cells, nuclei, organelles, etc. Okay? See you in class.